Hey everyone, I'm Patrick Jones, and welcome to episode 71 of That Gives Me Anxiety. Certainly it was an eventful day, I'll say that. I mean, I guess not really, right? But earlier, Ollie, the dog, barks and goes crazy, and then at the back door, and I go outside, and I see a bunch of baby bunnies. Not what I was expecting. Of course, I didn't let him out there. I don't know what happened to them. There must have been like I've been seeing like bunny poop around the backyard, so I figured that they were I had bunnies like I can call them rabbits too because I'm not seven. But when it's a baby, I guess it's a bunny. So I knew that they were there, but didn't know that there was like a nest or something. So yeah, I don't know what disturbed the nest, but I got there to two of the babies were dead, but there's one that's still alive. I didn't know where the nest was, but I tried to I put it in like some mulch and tried to like keep it together a bit, and I put some Spanish moss on top of it to kind of make it like a blanket. So yeah, I keep going out there to check on it. And I learned a lot about rabbits today. I've been Googling what to do in this sort of situation. Apparently the mom does come back multiple times throughout the day to feed. So fingers crossed, I'm gonna try to help this one rabbit out, this bunny out. I moved the two that died away because you know like we have eagles and hawks and snakes and stuff. So I was nervous that if something came to eat the dead ones would see the or smell the one that's alive, and so yeah, fingers crossed. Let's keep that little bunny alive. Other than that, Jamie and I had a great weekend. This weekend was the bridge run in Charleston. We walked. I mean, the race was 6 miles, a 10K, but walking from the car to the transportation to the finish line, and then finding because we were walking it, so our corral was pretty far from the starting point. So I think we ended up walking. I'd say more than 8 miles. Long day. I mean, we were both starving after, got a beer and some tater tots, and then both took a 3-hour nap. I mean, it's long, we got up at like 4:45. It's just a long day. But yeah, it feels good to do something athletically, even though my back hurting the whole time. Oh gosh. Well, enough about me and the bunnies and my back problems. I've got a great episode lined up. It's a little bit outside of the box. I interviewed Dr. Shelley Berger, who's a professor and a researcher in epigenetics. Yeah, I almost had a problem saying that word. It's from the University of Pennsylvania. If you don't know epigenetics, here, I'm just going to Google it. All right, from the Centers for Disease Control. Here's because I was going to give you a long-winded response. Epigenetics is the study of how your behaviors and your environment can cause changes that affect the way your genes work. Which is crazy, I guess I always thought that genes were locked in, but you could be doing things that alter how your genes work, and whether your body goes one way or the other. As it relates to disease, or I'll let the doctor explain that part. But the moral of the story is your genes are not set in stone, sort of like books that have already been written. Things like lack of exercise, your diet or exercise can turn on things you want to be turned on, or turn things off, which is pretty remarkable and interesting. And on top of that, I did ask the doctor about there's some research that suggests, or there's some research being done on whether or not traumatic experience can be passed down through genetics. So if you didn't get hugged enough as a kid, you might be able to pass that trauma down to your offspring. Again, listen to the doctor on that part. I was just interested to ask her about that. Just making sure I'm hedging enough here. Yeah, super interesting stuff that I really had no idea. and was so thankful to get the doctor's time to ask her these questions. So before we get to the interview, I just want to remind you, if you're liking the show, to please remember to rate and review it. 5 stars. What a great show. Boom. Send. You can check the show out on Facebook, Instagram, Twitter, and YouTube. Or YouTube. Check out my Minnesota Vikings hat. I hope it stays this purple forever. The purple is a big selling point. Also, a reminder of my partnership with Pure Spectrum CBD with the hashtag with the promo code AnxietyPod, which gives you 15% off. That link is in the description. Just click it in there, find the products that speak to you. I mean, I use so many of their products and always talk about it like it's just another tool in my tool belt to help with my mental health. And you can get 15% off with that promo code, AnxietyPod. And if you're liking the show and you want to place a tip, you can do that through the Buy Me a Coffee link. It's in the description as well. Well, I think it's time we go talk to Dr. Shelly Berger. As always, thank you so much for listening and enjoy. Joining me now on the podcast, I have professor from the University of Pennsylvania, Dr. Shelly Berger. Shelly, thank you so much for coming on. We're going to be talking about epigenetics today.
It's a pleasure.
Yeah. Yeah, absolutely. We've been playing a little bit of email tag back and forth, but I'm just thrilled because I really don't know much about epigenetics, and other than reading, so I'm up to date at least a little bit for our interview. But yeah, why don't you introduce yourself to the audience and give us a little bit of background of what you do and what you're working on.
Right. All right. So I've been a faculty member at University of Pennsylvania since 2009. And before that, I was at a small research institution. And I've been interested in epigenetics for most of that time that I've been a scientist, because it's a really fascinating area of study. And I'll explain that as we go along. So I study this topic of epigenetics in a whole series of really interesting areas, including aging and cancer and learning and memory, neurodegeneration, and ant behavior. And I can link all those together if we have enough time, but we'll pick and choose among those to discuss what epigenetics is and why it's so fascinating.
Oh my gosh. Yeah. All right. I'm so excited. Yeah. I think it's good to just start with a baseline definition of what epigenetics is.
Okay. So we can talk about it in a couple of ways. I'm going to start with what I think is the most intuitive way to understand what it is, and that is something that's familiar to all of us, which is the fact that we begin life as a single cell, which is the union of one sperm and one egg into a fertilized egg. And that fertilized egg then develops into the complexity of our entire body with all the tissues, all the cell types. There's about 200 different cell types. So if you think a little bit about that, you will fully understand that the same DNA information has to be present in every one of the different cell types in our body. So we start from one cell, that cell divides into 2, into 4, into 8, and off to the races. You generate 3 different major types of cell lineages, and each one of those lineages then develops into a whole host of different tissues. So we have in our bodies cells and tissues as different as brain cells and skin cells, liver cells, heart cells, and what have you. They all, this is the really crucial part, they all have exactly the same DNA. They share exactly the same DNA in every one of those cells. Same information, same genes in every cell. So what happens to generate all these different cells? Some genes have to be turned on or expressed, as they say in the business, and some genes have to be turned off or repressed. And the way that is done is through epigenetics. So epi actually translates epigenetics as on top of genes. So it's literally the regulation that occurs on top of genes.
Mm-hmm. That's a good baseline. What originally, it is just to reiterate and just underline what you're saying. It's just, It is so fascinating that one cell has all the information up to like creating a podcast, right? It's like all the way up. I had all the tools I needed, right? The liver cells, brain cells. It's like, it's incredible how complex the human body is.
Correct. It's that complexity that's so fascinating. So the DNA in each one of our cells, each cell is about 3 meters long if you stretch it out linearly.
Oh, wow.
And that DNA has to be packaged into a microscopic cell. Inside the cell is the nucleus. So inside the nucleus, inside the cell, the DNA has to be packaged. So that's an organizational problem, and it's also a functional problem because all the genes, there are about 20,000 genes in our DNA. The right genes have to be turned on, and the wrong genes for a particular cell type have to be turned off, so they have to be accessed. So the way this is done is the DNA is packaged much like a thread is spooled, is wound around a spool. So you think of, so we have proteins that serve as like structural, like the spool of a thread, a spool of a thread. So the DNA is wound around, and the proteins inside, the packaging proteins, are called histone proteins, and they become decorated by little chemical groups, and those little chemical groups either make the DNA bind more tightly to the spool or release it. When it's released, the genes that are there are turned on, are accessible to all the things that need to go on to turn a gene on and make protein. And when it's wound more tightly, the genes are turned off. Now, the epigenetic factors, and there are many, many of them, there's probably 5,000 different ways that are 5,000 different factors that add those chemical groups or take them away or do other things to change that packaging. So they are the epigenetic factors that carry out this process.
Oh, wow. You know, listening to you describe this process, it feels like a lot of aspects or characteristics of what ends up becoming of the result, right? The finished product are kind of left to chance based on the presence or lack thereof of a specific protein.
Okay, so that's a great question. How is it regulated?
Yeah.
It's a bit of a chicken and an egg question, because as cells from that single first cell that then moves on a trajectory to become brain cells or liver cells, those cells start to select epigenetic factors that then very specifically can find their target genes.
Oh, wow.
So I'll tell you a little bit about how some of this was discovered. I'll say that something else that I'm sure your listeners and you think about, if you think about science and human biology and biology in general, you think about mutations to genes as being a source of disease. Right?
Yeah.
There are about 20,000 genes. So, that's a lot of possible diseases of mutations. And you may inherit some of those from your parents. In fact, many of us think about that, that we're inheriting mutations that lead to cancer or to diabetes or to schizophrenia or to lots of other diseases. So that's right. There are a lot of mutations. But about a decade ago, a big breakthrough happened in biology. And that breakthrough it was even longer than that, about 15 years ago. New technology happened that allowed us to very rapidly sequence DNA. very high and very, very fast and very, very and pretty cheap. It used to be the first, the human genome was sequenced and it cost about $1 billion to do it.
Oh, wow. Yeah.
Now you can sequence a human genome for about, maybe a couple $1,000s at most.
Yeah, that's a long weekend in the Bahamas. Yeah, that's
Exactly.
doable. Yeah.
Exactly. And it's still expensive. I mean,
Yeah.
not everybody is going to do that, but it's doable. Especially for diseases. So what happened is a lot of sequencing went on of disease tumor samples from humans. And once these tumor samples were sequenced, like pancreatic cancer or lung cancer or brain cancer, now when we looked at where the mutations are, yes, many of them did occur in the 20,000 genes that make proteins, but most of the mutations, this was super eye-opening, did not occur in genes that make protein. Instead, they occurred between them. So genes that make protein make up only about 1% of our genome. Our genome is huge. The protein coding part is the genes is only about 1%. The rest of it is, serving various different functions. But what was amazing is that the sequencing of disease saw that many of the mutations were occurring in very small regions that regulate the turning on and turning off of genes.
Oh, wow.
And that's where the epigenetic factors that I was talking about a few minutes ago, that's where they function. They function at these small regions within DNA that regulate the turning on of neighboring genes.
And so that's exactly where the mutations were forming. Oh, okay. Wow, man.
This was incredible because all of a sudden epigenetics became a huge interest across, especially cancer. So, and then if we take a step farther, even the mutations in the genes occur in these, I was saying there's about 5,000. I don't know if that's high or low. I don't know. Maybe that's a little bit high, but even if there was 1,000 or 2,000, many of the mutations inside protein coding genes were occurring in the epigenetic factors that work at these little regulatory regions that regulate genes. So many epigenetic factors are mutated in cancer, and many of the places where they work inside the human genome are not at protein coding genes, but at the regulatory sites. So epigenetics just exploded.
Right. Yeah. My goodness. I mean, it's such a different, at least to me, right? I'm not in the field, but it's a different way of thinking about curing what ails us, kind of thing, right? Like it's a very different, it's a huge shift mentally in attacking the problem from a different perspective. So
exactly.
Does epigenetics, I'd imagine a large aspect of it has to do with...
Can I add one more thing before
absolutely.
you go? I just want to say that the epigenetic factors are often enzymes. And enzymes are kind of active proteins that do things. And you can inhibit them and activate them with small molecules. So there's a lot of work going out there in pharmaceutical world and the biotech world to find small molecule pharmaceuticals, drugs, i.e. drugs, that will activate or lower and increase activity or decrease activity of these epigenetic factors.
So that means if I have, let's say, pancreatic cancer, or possibly if I'm at risk of pancreatic cancer, you could theoretically, or is it in practice yet that you could tweak these DNA genomes?
Absolutely, it's in practice. Pancreatic actually turns out to be one of the really worst cancers, but many cancers, especially blood cancers, like leukemia. Leukemia is a blood cancer. It's a cancer of the blood that's circulating through your body, and drugs are much more. These tumor cells are much more accessible to drugs. It's much easier to get the small compounds to the tumor cells and into them. And they happen to have a lot of mutations in both the epigenetic factors and these little DNA regulatory elements that regulate genes. So there are quite a number of drugs now, either already in clinic or in clinical trials or in development, quite a large number.
Hey, that's very positive. Yeah. I had no idea 5 minutes ago that this was even possible. And, cancer has certainly touched my life and people I know. And so whatever works, whatever figures that out. I mean, that's amazing. Right. So how much of epigenetics, if this is possible to cure all these diseases in this manner, how much of it is?
I didn't say that. I didn't say that.
Okay, sorry, I said that. That's my bad.
Right. We're making some inroads, but there's a long way to go.
Yes, but I just meant that from the sense of like how much of epigenetics is focused on disease and curing that. Does it have other applications?
Are you talking about in academic research or in out there in the world?
In academic research.
Oh, well, you know what? One thing I'll say is that people are very interested in diet with respect to tweaking epigenetic factors as well, because there are various ways that some of the things that we eat can activate the good side with respect to, say, cancer. And lower the bad side. So, for example, I'll give you one example of a folate, which I think is vitamin B12, and it comes in through like avocados, that sort of thing. That folate activates a certain class of these epigenetic factors that is very good for suppressing. I think it's liver cancer.
Okay. Well, it makes sense why everyone's avocado crazy, over the last decade. Yeah.
Right. Avocado oil that's.
Yeah, absolutely.
Yeah. So there's an example of diet. The other thing I'll say is, have you learned about or interviewed anybody about the microbiome yet?
No, I haven't.
Okay, this is a fascinating area of research where it turns out that your gut, all of our guts have a certain flora and fauna, animals and plants, microorganisms that are living in our gut. It's quite diverse. And you can have a good microbiome or maybe a deficient one, depending on what you eat and what whether you exercise. Exercise is superb for anti-aging and good health. So all of those things impact your microbiome, and your microbiome, it generates naturally small molecules that alter the activity of some of these epigenetic factors. So that's another kind of interesting sidelight of the way current research, very cutting edge type research.
Yeah. Can we talk? I know you've got a lot on your plate. You've got work, you've got friends, you've got family, pets, you've got the people that you make small talk with at the coffee shop or gym. You've got that bird that you see when you wake up every morning outside your window that you've projected things onto. Look at that bird. Doesn't even love its family. It's always by itself. You do that. Everyone does that. Point is, you've got a lot on your plate. Well, that's why there's Instacart, to take a little bit off your plate. Using Instacart, you search for all your favorite foods and things that you need from the grocery store, all online, all while you're looking at that bird, wondering why it hasn't called its mom. And they deliver it to you. They go to the store and do the shopping for you. And they can deliver it in as fast as an hour. And you can sign up by clicking the link in the description, wherever you're listening or watching. And that's a great way of supporting the show. So it's a great way of supporting this show. It's a great way to make your life a little bit easier, because we all know that you have so much going on, like wondering whether that bird judges you back. So in listening to you describe this field, I'm kind of caught in between. Doctors often ask you who in your family has had cancer, right? Or predeterminations, right?
Mm-hmm.
How much of do you see where I'm going with it? That it's like you're describing these mutations that somewhat feel random. Is that information passed along from your parents?
It absolutely can be the same as, very similarly to the way you think about a mutation in, I'm trying to think of a common one that doesn't have anything to do with epigenetics. Well, you know about breast cancer and BRCA1 and BRCA2. Those are really famous ones. Those are the ones that Angelina Jolie had one. And that was the reason she, announced, she wrote columns about her mastectomy, double mastectomy and hysterectomy, because she had a mutation in something that mutation leads to a problem in repairing your DNA when it's damaged. So that's a different sort of mechanism. And that's the kind of thing we commonly think about. But, mutations in these epigenetic factors can also be inherited. So similarly to non-epigenetic factors.
Yeah. Oh, wow. In doing some research, and if this is outside the realm of what the subject matter actually does, please tell me. I saw certain studies that were talking about how traumatic experience could be passed down through some manner down to our cells. Is that accurate? And if so, I'm curious.
Yeah.
To learn more about that.
So, right, this is an area of epigenetic research. You're right. So it's pretty controversial.
Right.
I mean, there's evidence. It's very difficult to do these type of studies. Let me explain it a little bit. So what this is called is transgenerational inheritance. And what this means is through diet or through parental care, various things that are transmitted through the offspring, but not through genetic mutations, right? That's what you're asking about. You're asking about various things. So it could be something as tangible as diet, or it could be one thing that's been studied a lot is the licking behavior of mom mice to their pups. So that's kind of like a equivalent of a nurturing kind of behavior. The way moms and dads, or moms and moms, whatever, the parents of kids, hold their child, talk to them, interact with them. That would be the equivalent. We don't lick our kids, but that's what mom mice do.
Okay.
And so the idea is that the equivalent would be, if you didn't give that to your child, maybe that would be abusive, if to a certain level.
Right.
And the idea is that there are changes that occur in the brain of the pups in response to the licking or in response to our nurturing that will change the epigenome.
Oh my gosh.
So now I'm talking about changing those little regulatory elements, changing the way I was telling you about the way these histone proteins that package DNA, they get decorated with chemical groups to open and close and turn on and off genes. These little regulatory elements in DNA, they can also be decorated with chemical groups. And it's kind of famous, called methylation. And this methylation can lead to the turning on and off of neighboring genes.
Oh my goodness.
And so that's the idea. The data supporting it is, it's hard to completely understand the mechanism of how the licking behavior gets translated into very specific mechanisms, very specific endpoints of the methylation of certain DNA regulatory elements.
Yeah, it would be hard to eliminate variables, I'm sure, right? It's so many other complicated things to isolate that.
That's exactly right.
Yeah.
That's part of that. You actually, what you say, Patrick, is exactly what science is all about. Having the right control experiments so that your experimental paradigm is meaningful. And it's hard to do that in certain experiments. And this is one of them, this kind of transgenerational exploration of these behaviors.
Oh.
I mean, the other one that the data are coming from, something that's a little bit easier to follow now, is parental diet.
Mm-hmm.
So the experiment that was done is that the father, now we're talking about mice,
Mm-hmm.
the paternal mouse, the father, was given a diet that was very either rich in protein or low in protein. And then so the control was the normal protein level of a mouse diet. And then the experimental was, I don't know, I can't remember if it was low or high protein, but whatever. And so that now the mice were mated to females, and the females had offspring, and then their offspring have offspring. And so that's called the first set is called the F1 generation, and the second one's called the F2. And by the time you get to the F2, because you started with the male mouse, it's not, and I'll just say that most of the factors that come through a mammalian mating come from the mom, because the egg is big, the sperm is tiny, and almost the only thing the sperm gives is DNA, whereas the egg from the mom gives DNA and all kinds of other factors that get the fertilized egg started and dividing up to a certain time. So in using the male as changing the diet of the male rather than the mom, the dad rather than the mom, that allowed, by the F2 generation, there was nothing left. It was only through the genome and not through anything that was directly transmitted. Do you see what I'm saying?
Yeah. Wow.
With these transgenerational experiments, you have to eliminate any direct transmission through the egg and the sperm. So by the time you get to the F2, you're pretty confident that you're looking at changes that aren't mutations now, but are changes that are occurring epigenetically, are happening because of that diet, either the normal diet or the stressful diet. And by the F2, you can see that the metabolism of the offspring altered. And
wow.
Yeah, it's affected. So the liver, you can look at the liver and look at the metabolic profile. The liver is the metabolic organ in the body. You can look at the way the liver functions and whether important metabolic factors are high or low. And this group could see that there was an alteration. It was carried through to the F2. So it was epigenetic.
Oh my gosh. That's incredible. That's so wild, right there. Yeah. I'm just going to throw a blanket statement of like, man, we just know so little about, about our world,
exactly.
right?
Exactly.
Oh my gosh.
But that then opens up the possibility that many different aspects of our diet, male and female, women always say, oh, you got to eat or not drink or blah, 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 blah. But it turns out what I'm telling you is that stuff can come through the male from the sperm as well as the egg. So
and need to keep eating well as well, I guess, if
exactly. And by the way, that may explain. So another area that maybe you're interested in or you've talked about in your podcast is the rise in autism. And part of that maybe is linked to the age of dads when they have kids, that you can see that sperm from older males is altered in various ways.
Wow.
And the DNA is altered. So that could be due to that. I'm not saying that's epigenetic necessarily, but I'm just commenting on the fact that
it's possible.
Yeah. Eggs age, but so do sperm.
Right. That's true. What drew you to epigenetics?
From the time I was really an undergrad, I've been interested in how what regulates genes. To me, it seems like a fundamental question when I explained right at the beginning that when you think about the fact that we develop from one cell.
It's crazy.
Mind-boggling, and how often it goes right is amazing. I mean, it does go wrong. Obviously, we know it goes wrong sometimes, but the miraculous fidelity of that process is truly amazing. And so that means that with unbelievable precision, genes have to be turned on and off at the right time and exactly precisely to get perfect development. Mm-hmm. And that goes from the simplest multicellular organisms to the most complex.
Yeah. Oh my gosh.
Really a fundamental question. So it intrigued me from the beginning. And when I was a postdoc, I identified with my advisor at the time, kind of a new set, not previously known, a new set of factors that are involved in turning genes on and off. Okay. Mm-hmm. So that was cool. Then I started my own lab. Okay. And then it turned out I didn't discover this, but one of the factors that I discovered, someone else realized that it's one of these epigenetic factors that was able to put on these little chemical groups that I've been telling you about today. Mm-hmm. And so that was like amazing, because before that time, almost everyone studying this broad field of gene regulation, what turns genes on and off, mm-hmm. had been studying all kinds of different factors that were involved in the mechanics of turning genes on and off, but no one was studying this set of all of these epigenetic factors. We didn't even know most of them. So this started about, we're going on 20 years. Mm-hmm. It kind of like exploded a new field of understanding the relationship between turning genes on and off because of what I had discovered, and the finding that one of these factors had this property of being able to change the epigenetic landscape of genes turning on and off.
Yeah.
And I'll just say that I then spent my lab's been studying that ever since, mm-hmm. in many different contexts.
Right. Yeah. I mean, there's innumerable ways to go about that, right?
Exactly.
Yeah. I mean, you're basically cracking into where we come from, right? Like there's like IKEA directions in our cells, and
exactly.
It's like what
I like that. Mm-hmm.
Yeah. It's exactly. Like who put it there? How did it get there? It's just it's incredible. It's fascinating.
Exactly.
One of life's mysteries is that birthdays always find me unprepared, catch me unprepared. I don't go on Facebook as much, so I don't notice them as much. But I want to express my love and appreciation for friends and family. That wasn't part of the copy. I'm just saying it. Year after year, month after month. Even when I see it coming, I'm not ready. Am I a jerk? Yes and no. Why do things have to be so black and white for you? But I've got great news, friends. Introducing monthly card subscriptions from the Cardist Studio. Join the service that delivers the card and the stamp that you'll need to your door. No more errands. Who wants more errands? I don't know why, but I'm starting to think of errands as a kid's name. Errands, go back to bed. Okay, but here's how it works. You choose your categories, from birthdays and celebrations to love and encouragement, or select their full collections and have those thank you notes and just because cards at the ready. Those ones are always so nice. Like when you get a card for doing something and it's just like a small little heads up or token of appreciation, it genuinely makes you feel really good. And best of all, you would never get caught cardless again. Let me repeat. You'll never get caught cardless again. Try to say that a bunch of times in a row. You'll never get caught cardless again. Monthly card subscriptions from as little as $8 per month for the card and the stamp. Delivered to you. TheCardistStudio.com shop subscriptions. And you can use the promo code AnxietyPod for 15% off your originating order.
So that was pretty cool. And then my lab studied a lot of the mechanisms. We discovered new new epigenetic factors for about 10 years. And then all of a sudden I decided, I want to study this in the context of real physiology. Like, how is this all used? Not just turning genes on and off. That's a cool question. The mechanics of all of that. But where is it really important? And so I started to study this in the context of aging. That was the first area I went towards. How are these epigenetic factors? How are they? Of course, it's a huge question. Why do we age? Why do our cells age? What goes wrong? And there are a lot of different things that go wrong. I'll say that. But one big area, and my lab helped to discover this, is that epigenetic. There start to be changes in key epigenetic factor. A few of these key, probably a large number of them, but at the time studied one key epigenetic factor that it's level. It's an enzyme. It removes one of these chemical groups that I was telling you about. It removes it, and its level drops precipitously during aging.
Okay.
And because its level drops, this chemical group that it takes off, it remains very high, or it even elevates as we age. And that screws up some key parts of our DNA.
Oh, wow.
For example, the ends of our chromosomes have to be very compact. Otherwise, if they're not compact, they tend to, you get mutation, you get chromosomes that join, and that creates big problems in cell division and that sort of thing, and leading to cancer and other problems. So not the high level of this chemical on this chemical group on in these epigenetic regions at the ends of chromosomes leads to aberrations, leads to problems, dysfunction of the telomeres, the ends of chromosomes. So that was a big finding that really opened up epigenetic research in aging. So, my lab is recognized for helping that. Now it's a huge area of research. A lot of people are studying.
A lot of money in helping people age gracefully, or I guess not at all, right? I mean, if people could live to 500, some people would choose to do so.
They would. Yeah. And there's a lot of money, and a lot of very wealthy people have invested in the area of aging. So aging research, I mean, all aspects of it. I'm not talking about only epigenetics. There are a lot of different areas to study. So that's one area. The other area I became really interested in, and it's a little circuitous how I got there. I won't explain it, but learning and memory. And we studied that in mice and humans. We're interested in what promotes the ability to learn in our neurons and in our brains, what happens during aging and during disease, like neurodegeneration, like Alzheimer's disease, and what epigenetic factors are involved in robust learning and memory, and then on the other side of the coin. Well, and in aging well, which is a great thing. I mean, no matter when you die, you want to live to that point with your brain intact, right?
Absolutely. Yeah.
So what maintains cognitively, like what is it in humans, in cognitively robust humans? What kind of epigenetic things are at work to maintain a healthy brain? And then what are some of these things lost in Alzheimer's disease? So we have, to study that, we have gotten, there's a biobank here at Penn with cognitively normal people who've donated their brains to the biobank, cognitively normal and Alzheimer's disease. People who've died, sadly, of Alzheimer's disease, they've donated their brains. And then we want to compare to, very sadly, young people who have died for whatever reason, accidents or whatever, and have donated their brains. And so we'd like to compare young, robust, cognitively aging humans that are, really robust, and then Alzheimer's disease. And so we found that some epigenetic factors that I've been telling you about actually get increased in cognitively normal brains with aging. So we think they're protective to brains, to neurons, to preserve them and make them our neurons in general. We're born or early development. We have most of the neurons we're going to have our whole life. So obviously maintaining the robustness of those neurons is absolutely crucial.
Right. I wash my car and I'm not going to have that as long as I'm going to have my brain. Right. So
that's right.
So you
want to keep your brain washed. Right.
Yes.
Exactly.
Oh man, what a fascinating job you have. My goodness. Yeah. It's so interesting, right? A lot of people have jobs that don't fulfill like the creative and thirst for knowledge, right? That, I mean, you're right on the cutting edge of understanding humanity. I mean, that's
well, I don't know about that, but one aspect, there are many aspects of humanity. So
yeah,
it's one aspect, and I totally agree. And giving something to your listeners, scientific research, a lot of people think it's really too difficult for average person to do, and that is not the case. It's really fun, and what you learn in grade school and high school tends to turn people off of science, but it turns out that doing experiments, if you're trained, you go through the stepwise training, you end up being able to do really, really great, really fun work that hopefully will benefit humankind.
Absolutely. Yeah. That's certainly a great goal.
If you have another minute, it's not only human biology that we're interested in the world. We also study ant behavior.
Why ants?
Yeah, ants are absolutely fascinating epigenetically because it turns out that in a colony, there's a queen. So ants are much like bees. There's one queen, and she's carrying out all the reproduction. And then there can be 100, depending on the species of ant. There's 100,000 species or something. Depending on the species, there can be 100 or there can be literally millions of worker ants. The worker ants are also all female.
Are they?
All female.
I didn't know that.
Soldiers, foragers, all the, nurses, they are all female. So when you look at a typical ant colony or an ant nest, they're all female. There are males, but they live a very, very short time. They inseminate, they fertilize the queen, and then they die. In addition, the whole system is a great aging question epigenetically because the workers live about a year in most species. Queen can live 5 to 30 years.
30 years?
Yes.
Holy cow. That's crazy.
Right. So this is all determined epigenetically. All the females share pretty much the same genome. Again, it's exactly analogous to the cells in our bodies. The cells in our bodies have all the same DNA. And remember the beginning, turning genes on and off to make different cells in different tissues. Ants are the same. They're all female. And depending on which genes are turned on and which genes are turned off, they're going to end up being foragers that go out and find food, nurses that stay home and raise the new brood, the new ants, or soldiers that protect the colony, or queen. And all she does is lay eggs. So we're very interested in the learning and memory, the learning part, the behavior part. How does this happen? And we think it's translatable to humans, a lot of it, and the aging side as well, through trying to understand the plasticity of the ant brain, because we can convert soldier to forager.
You can.
Using epigenetic means, using epigenetic small molecules, these little drugs I was telling you about, we can inject them in the mouse brain and change a soldier into a forager. So we're trying to understand how that occurs, and it's restricted. It can only happen in very young ants right after they're hatched. And so it's sort of analogous to humans, that our brains are plastic when we're young up to a certain point. And of course, our brains become less plastic as we age. So we think there are really interesting translational things. So the point being that epigenetics is an interesting concept, not only in human biology and human disease, but trying to understand biology, you know?
Yeah, absolutely. Yeah. Well, I've really enjoyed this conversation. I want to make sure, I mean, we could go on and on for weeks. But is there anything that you think that is almost vital or very important that someone who wants a baseline understanding should know?
I think it's really this idea that the concept of epigenetics, we're so married to the idea of genetics, and we are our genetics, we are our genes. And it turns out that the environment, all of these forces, we really didn't talk about the environment, but our diet, our microbiome, all of these aspects of how we behave, what we eat are affecting our epigenome, and that's affecting us in many ways. Oh, and I'll also add exercise. It definitely affects our epigenome as well. So very important to do that. So, that, I guess that's the main message to leave you with, leave listeners with.
I mean, that's a big shift, speaking personally, right? Because I always pictured my DNA as like set in stone, and really it isn't.
Exactly.
And you need to keep.
It's malleable. It can be altered. Exactly.
Yeah. Wow. Well, Shelley, this has been an incredible conversation. I've really enjoyed speaking with you, and thank you for being patient with me as this was certainly a little bit above my pay grade. So
navigate unfamiliar territory.
Yes.
Well, it's been a pleasure to talk with you as well, Patrick. And I'll look forward to listening to some of your other podcasts.
Yeah, please do. Always open to new ideas if you ever have any, or feedback as well. Yeah.
Okay.
Oof. Was your head spinning as much as mine was? Well, trying to hang on for dear life. Shelley is... thank you so much to Shelley. She does such a good job of explaining it and staying patient with someone who is way behind her in intellect. But I want to be. I want to learn. Things like this are just so hard. But I guess I should cut myself some slack. It's an advanced, cutting edge thing that she's researching, and I'm Pat. Well, yeah, thank you so much to Shelley Berger. Before I get to the weird thing causing me anxiety this week, I just want to remind you of the Buy Me a Coffee link. The link is in the description, and it's just a way to make a tip or a donation to the show. Then there's the Pure Spectrum CBD with the promo code AnxietyPod, which is also in the description. You can click that link right in there, punch that in, get yourself some gummies or some bath stuff or some Solvays. They got whatever you need. I'm telling you. If you want to sign up for home delivery of groceries, you can do that through Instacart, and that link is also in the description. And then the Cardist Studio is offering the promo code AnxietyPod for 15% off your initial order. So yeah, supporting those sponsors helps support the show and helps me keep making these. So thank you to them, and thank you to you. Okay, so the weird thing causing me anxiety this week, besides from, I'm still settling into the anxiety of keeping this bunny alive. I just saw a big bird as I was reading, and I had to stop for a second because I was like, is that bird gonna go near my bunny? But then I'm choosing the bunny over the bird. The bird still wants to be alive. Don't get involved, Pat. I'm involved already. But it's taxes. I always have my taxes done by someone else, but I haven't heard back in a week since I've sent everything in. And so I've just been dealing with this paralysis of oh God, do I owe money? Please don't let me owe money. It's just, you're just sitting in this limbo of something really bad could happen or something good could happen. I could get a few bucks back, which would be always helpful. We are trying to get married here. But the state of unknowing, I'm constantly having to distract myself from being oh, did I get the email? Did I get the email? Nothing yet. So I'm just going to stay stiff as a board until I find out. Well, I'm rooting for you with all your taxes. I hope you Big money, no whammies, right? Well, as always, thank you so much for listening. Have a great week, and I'll talk to you on Thursday.